Okay, so the first part of this lecture will be on understanding how DNA is organized in the cell, and one of the basically background right here that you also need to make sure that you understand is the cell cycle. So mitosis and meiosis are the subject of this lecture, but mitosis and meiosis are just a very small part of the total cell cycle. So the interphase refers to the phases of the cell cycle that don't include mitosis and meiosis, and the interphase is broken up into multiple phases, so G0, G1, S, and G2. So the cell is sitting around, and perhaps it decides that it wants to divide into two different cells, and so it has to grow, and then it has to synthesize its DNA. It has to replicate many of the organelles that are within the cell. It has to add cell wall. It has to create new ribosomes. And this whole process requires a lot of energy and a lot of growth, but it ends with two cells that have their own DNA content. And so the cell cycle includes these G1 and G2 phases, which are growth phases, so the process of the cell actually getting larger. And then importantly, there's this S phase that's in between these, and this is actually when DNA is synthesized. So every DNA molecule within the cell is copied by DNA polymerase. And at this point, it's pretty much distributed throughout the cell. And even after S phase has occurred, all of these DNA molecules are pretty unorganized within the cell. What happens during mitosis or meiosis, which is in between the G2 and the G1 phase, is that the DNA then gets organized into chromosomes, and the DNA is segregated into the two different cells. So mitosis and meiosis just refers to that phase of separating the DNA, and it's important to remember it's not when the synthesis of the DNA actually occurs. So if we actually took cells in this phase and we looked at their DNA, we would be able to see these things called chromosomes. So karyotype analysis is the process of being able to take a photograph of chromosomes in a cell. So typically these are taken from blood cells, fetal skin cells, or bone marrow cells, which are actively dividing and in mitosis. And so if you follow these steps, so if you identify these dividing cells that are in mitosis, if you lyse the cells to release the chromosomes from the cells, if you stain the chromosomes using dyes that target DNA, and then you sort the chromosomes by length and homology, you'll get a karyotype that basically looks something like this. And so this is an example of a karyotype from humans, and you'll notice that there are 23 chromosomes. So humans have 23 unique chromosomes. 22 of these are autosomes, and then one of these is the sex chromosomes, which can either be the X or the Y. And depending on your sex, you either have 2X or an X and a Y sex chromosomes. But one of the things you should notice is that each individual has 2 copies of each chromosomes. So there's 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, and that's because we are diploid. So this is part of what we mean when we're diploid. When experimenters were first doing karyotypes, they were able to determine that we had 2 copies of each of these chromosomes within our cells. And so this represents a chromosome 1 that you received from your father, and then this represents a chromosome that you received from your mother. So again, you receive one of each chromosome from both of your parents, so you have 2 of each copy. So, you know, the experimenters basically see this garbled mess of DNA, and then they have to sort these into this nice looking karyotype. And so the way they do that is using size criteria. So the length of the chromosome is obviously very important. So you can see all of these chromosomes have different length, and they're sorted in the order of the longest to the shortest, except for the sex chromosomes. And you can also see that the DNA stain actually stains different parts of the chromosome to a different extent. So there's a very bright locus on the tip of chromosome 1, for example, and that could be a criteria for how you look through this mess and then figure out which one is actually chromosome 1. So you can look at this and say, oh, this is chromosome 1 and this is chromosome 1, and then you can cut it out and kind of move it into the proper position. And so if you're an experimenter that was doing this, you would just have to know the DNA banding pattern that you would expect for each chromosome and the relative size, and then you'd be able to create something that looks like this. So this is a karyotype. So basics of DNA organization. So chromosomes were discovered in 1843, and they can actually be seen using light microscopy in certain cells. So these things are large enough and opaque enough that you can actually see them just simply with light microscopy. But they're not seen until after DNA replication has occurred. So again, DNA is normally very disorganized within a cell, and if you stain DNA, you would not be able to see these very nice structures. So chromosomes are actually highly compacted forms of DNA. So your DNA is normally compacted within a cell just to fit within the cell, but during mitosis and meiosis, further compaction occurs to really condense these down into very compacted forms. And then studies showed that DNA is organized into multiple chromosomes. Some chunks of DNA were very similar, which were revealed to be homologous chromosomes, and then humans and most animals are diploid, so there's 2 homologous copies of each. And this led to a definition of the diploid number, where the diploid number is the number of chromosomes that are seen in a karyotype. So if we counted these up, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc., we would end up seeing 46 total chromosomes within the cell. And so that is what the diploid number is for humans. So the diploid number of humans is 46, and it's the number of chromosomes, and it's also often defined as 2 times n, where n is the number of unique chromosomes. So there's 23 unique chromosomes. So there's a chromosome 1, 2, 3, 4, and then the sex chromosomes are kind of lumped together. So there would be 23 unique different chromosomes, and then because we have 2 copies of each, our diploid number is just 2 times the number of unique chromosomes. So a chromosome, this is showing you a single chromosome right here. So if you look at this structure right here, what this is actually matching in the karyotype is something that looks like this. So this can be a little bit confusing at first, I think. But a chromosome is not just DNA, it's actually composed of both DNA and protein. And it's actually an important thing to remember. It's actually composed of 2 DNA molecules. So there's not just a single DNA molecule within a chromosome, there's actually 2. And that is because DNA replication has occurred, and so you actually have 2 exact copies of your chromosome 1, and this would both be, for example, from your father. So the cell initially has a single DNA molecule from the father, DNA replication occurs, and now you have a new copy of the father. And so that's something that's important to remember. So a chromosome actually has what's called sister chromatids, and so these sister chromatids are the 2 DNA molecules. And these are actually held together by these proteins. So in the middle you can see that there's a bunch of proteins that kind of hold these things together. So cohesin is an important protein, and then sugarshin is also an important protein that kind of acts to hold the sister chromatids together. We are going to talk in a little bit about how these are actually separated into 2 different cells, and that involves the attachment of microtubules. And so the spindle fibers of this actually connects to the chromosome, but it's actually at a very specific point. And so typically that's towards the center of the chromosomes, and this region is called the centromere region. So the part of the DNA that actually is where the spindle fiber binds and the kinetochore actually forms is called the centromere region. And so this is actually what causes that kind of characteristic shape of a chromosome, where very typically you draw it looking something like this. And so that is because this centromere region where the spindle fibers attach is pinched in upon each other, and so that causes it to look like that. And so this, for example, again, would be one of the sister chromatids. This would be one of the DNA molecules. So obviously it's wrapped up a whole bunch, but if you unwrapped it, you would end up having a single DNA molecule within here, and then a DNA molecule within here. So again, chromosomes are composed of both DNA and proteins, and centromeres are the regions where the spindle fibers attach during meiosis and mitosis in order to separate these things out. Some other important terminology, telomeres. So telomeres are the ends of the chromosome. So this would be, the telomeres would be here and then here. And then chromosomes are named based upon the position of the centromere. So one of the naming systems that's used is dependent upon where exactly the centromere is located. So it might be directly in the middle, and then so if it was in the middle, it would be metacentric. The other extreme is that it's almost at the end. So if this part right here was moved up, kind of how the spindle fiber attached and where the centromere was, then you would have something called telocentric. And so telocentric comes from, telomeres, kind of that's the shared origin of the word. And then there is submetacentric and agrocentric, depending on different extreme levels of how far apart they are towards either the center or towards the end. So if they're close to the end, they're agrocentric, and then if they're between the middle and the end, they're submetacentric. It's kind of a loosey terminology. Another thing that's important to remember, so you might actually start to see gene locations, and you might actually start to see regions of a genome that are implicated in disease, and very often you'll see something that starts, a location that starts with like a 2P, a 3Q, or something like that. And so this P and the Q, where that comes from, is that the location of the gene could be on chromosome 2, chromosome 3, and then you can further indicate the location of a region of the DNA based upon, is it on the short arm, which is the P arm, or the longer arm, which is the Q arm. So that's a further way that you can kind of define where DNA is whether it's on the short arm or the long arm of a chromosome. All right, so human chromosomes. So this is just taken from Wikipedia. Again, human diploid number is 46. We have 23 unique chromosomes, and if you wanted to see how long each chromosome was, you can see this in this table. So this nice table lists both the approximate number of genes that are on the chromosome and then the total length of DNA. So chromosome 1 has approximately 2,000 genes, and it's about 250 megabases long. So pretty long piece of DNA. So chromosome is pretty long. And so you can see in general things get successively smaller and smaller and smaller. The exception is actually chromosome 22 is actually a little bit bigger than chromosome 21, and so the initial naming was based upon the karyotyping, and so they didn't exactly get the size of it right, but overall they're pretty accurate. And so if you add this all up, and then you have the sex chromosomes, the X chromosome, and the Y chromosome. So the Y chromosome is actually a little bit larger than 21 and 22, and then the X chromosome is one of the larger chromosomes, kind of in between the 7th and 8th. If you add all these up, you have about 21,000 genes. So the human genome encodes about 21,000 genes, and there's about 3 gigabases of DNA that are encoded. Chromosome organization is unique to each species, so it's evolutionarily pretty labile, which might be surprising. So for example, if we took this beetle right here and did a karyotype on this beetle, it would look something like this. And so you should be able to count. Okay, so the first thing you should notice that it's diploid. It is diploid because there's 2 copies of each chromosome, and then there are 12 autosomes and then 1 sex chromosome. And so if you counted all these up, the diploid number of this beetle would be 26. The salamander right here, the diploid number of it is 32, and then this bird right here actually has more chromosomes, so there's 32 autosomes, and then actually they use a different sex determination system, so there's a ZW chromosome, it's called in birds, but there's 66 total chromosomes in here. And so this is just a table, again, that kind of shows the extreme distributions of chromosome number. And so again, this is defined by karyotypes, and you use the karyotypes in order to count how many chromosomes and then determine is it diploid, triploid, etc. All right, so homologous chromosomes. So homologous chromosomes, if we wanted to look in this, we would say this chromosome right here is homologous to this chromosome right here, and so on. And so we have just shown this this schematic of a karyotype. So you see these stains. So this is kind of the idea of this is what a DNA stain would look like. And so homologous chromosomes are very similar to each other. So they have pretty much usually the exact same size. They usually have the exact same banding pattern. And I say usually because there's some genetic disorders that are known to actually change this. But normally these chromosomes would be the exact same size, and they would have the exact same banding pattern, and they'd also have the exact same centromeric position. So, and then again, the idea of this is that one of these came from your mother and one of these came from your father, and that's why you have two. So these are not identical to each other, so they're very similar in sequence, but if you actually started to read the DNA sequence of this, you would find that there were differences, and there's not a lot of differences, and it's along the lines of every 1,000 base pairs. So if you read 1,000 base pairs, on average there would only be one difference from the chromosome from your mother and then one chromosome from the chromosome from your father. But you go back to this table, that is actually a large number of differences. So if we did 1 over 1,000, that means your mother and your father's chromosomes, chromosome 1 differs by approximately 250,000 different base pairs. And this can actually have very important consequences on phenotypes. And that's what a large part of this class will be to study. So it's important to remember homologous chromosomes, one comes from your mother, one comes from your father, but they are slightly different from each other. So I talked a little bit before about you could also have triploids or tetraploids, and so this is an example of a karyotype from that. So you can see that it's kind of hard to tell. You might need to be a professional karyotyper in order to really be able to build this thing, but this is an example of a triploid organism where you have 3 copies of chromosome 1, 3 copies of chromosome 6, etc. And then this is an example of a tetraploid individual that has 4 copies of each chromosome. So all 4 of these would be homologous chromosomes. And so that's how you would read the karyotype in order to kind of distinguish, what the ploidy is. So this comes from a squash, and I'm not actually sure what the species is anymore. All right, so the final thing to talk about is the genome. So the genome is one final term that I talked about at the beginning of lecture that you need to know. And this is a little bit tricky at first. This is the DNA content of a haploid cell. So when we talk about the human genome was solved back in 2003, I think, and if we want to download a file, we can have a file of the entire human genome. So you can go to NCBI and you can actually download a text file that contains the entire set of nucleotides for each chromosome. And so it looked like something like this. It's called FASTA, but you would have chromosome 1, and then you would go on and list, approximately 250 million base pairs, kind of over and over again. So when we talk about the size of a genome, we are talking about the amount of DNA that's in a haploid cell. So this file does not contain 2 copies of each chromosome, because if you looked in a real cell, you would have your chromosome 1 from your mother and then your chromosome 1 from your father. So if we had the amount of DNA within a cell, then you would have to list both of these. But that's not actually what's included in a genome in the concept of a genome. So the concept of a genome is the set of DNA that is unique. So you would have one chromosome 1, one chromosome 2, one chromosome 3, etc. And when we get into meiosis, you'll see that's actually the exact amount of DNA that is within a haploid cell. So a sperm cell or an egg cell, an oocyte, those both have only one copy of each chromosome. So you can think about it as a genome is the amount of DNA that's within a haploid cell. All right, so these are just some of the definitions that I include for your convenience, but hopefully kind of over the course of this lecture, you've come to understand genome, DNA molecule, chromosome, chromatid, homologous chromosomes, and then sister chromatids. But you can read up on the definition to make sure you understand that. All right, so that's the end of part 1.